Assalamu alaikum and hi to everyone today. Our group will be presenting on the formation of Koilocytes, HPV-induced cellular changes, and I will begin our presentation with an introduction to human papillomavirus, or HPV. Human papillomavirus, or HPV, is a small, non-enveloped, double-stranded DNA virus from the Papillomaviridae family. More than 200 HPV genotypes have been identified. HPV infects basal epithelial cells through tiny breaks or microabrasions in the skin or mucosal epithelium. It mainly infects the cervix, anus, vulva, penis, and oropharynx. Most HPV infections are temporary and are naturally cleared by the immune system within 1 to 2 years. However, persistent infection with high-risk HPV can lead to abnormal cell changes and eventually cervical cancer. This slide compares low-risk and high-risk HPV low risk. HPV includes types such as HPV-6 and 11. These viruses have low oncogenic potential and are commonly associated with genital warts and low-grade squamous intraepithelial lesions, or LSL. They usually remain episomal, meaning that DNA does not integrate into host genome and cleared by the immune system. In contrast, high-risk HPV includes types such as HPV-16 and 18. These viruses have oncogenic potential and are more likely to integrate into the host genome during persistent infection. They are associated with high-grade squamous intraepithelial lesions, or HSL, and invasive cervical cancer.
This slide explains how persistent high-risk HPV infection progresses to cervical cancer. On the left, the cervical cytology progression starts with high-risk HPV infection. Persistent infection first causes LSIL, which shows koilocytic changes. If the infection is not cleared, it progresses to HSIL, and eventually to invasive cervical squamous cell carcinoma. On the right, persistent HPV infection allows viral DNA to integrate into the host genome. This causes overexpression of the E6 and E7 oncoproteins, which inactivate the tumor suppressor proteins P53 and PRB, leading to uncontrolled cell proliferation in cervical cancer. This slide explains why cell death evasion is important in HPV infection. Normally, apoptosis, cell death, removes virus-infected and damaged cells. However, HPV prevents this process using 3 oncoproteins. E6 promotes the degradation of P53 and activates telomerase, allowing infected cells to survive longer. E7 inactivates PRB, causing uncontrolled cell division. Finally, E5 suppresses apoptosis and helps the virus evade the immune system by reducing MHC class I expression. Together, these mechanisms allow HPV-infected cells to survive, accumulate genetic mutations, and increase the risk of cervical cancer.
Before we understand how HPV causes koilocytosis, let's first look at the normal structure of the cervical epithelium. The cervical epithelium is made up of 4 layers: the basal, parabasal, intermediate, and superficial layers. The basal layer contains keratinocytes that continuously divide to produce new cells. As these cells move upward, they become more mature through a process called differentiation. Eventually, they reach the superficial layer and are naturally shed. This is important because HPV only infects the basal keratinocytes, as these are the only actively dividing cells. So, how does HPV enter the cervix? HPV cannot infect healthy, intact epithelium. It needs small injuries called microabrasions to reach the basal layer. These tiny injuries can happen during sexual activity. Once the basal cells are exposed, HPV attaches to the basement membrane and enters the basal keratinocytes. The viral DNA then moves into the nucleus, where it stays as a circular DNA called an episome. At this stage, the virus establishes infection without killing the cell. After entering the basal cells, HPV begins its life cycle. In the basal layer, HPV DNA stays inside the cell. The viral proteins E1 and E2 help copy and maintain the viral DNA while the infected cells continue to divide. As these infected cells move upward, HPV becomes more active. The virus produces E5, E6, and E7, which keep the cells dividing and help the virus make more copies of its DNA. When the cells reach the superficial layer, the L1 and L2 genes produce the viral capsid proteins. New HPV particles are formed and are released when the superficial cells are naturally shed. This table shows what happens in each epithelial layer. In the basal layer, HPV starts the infection and expresses E1 and E2. In the parabasal layer, it produces E5, E6, and E7, which help keep the infected cells dividing. In the intermediate layer, the virus makes many copies of its DNA. Finally, in the superficial layer, L1 and L2 are produced, new virus particles are assembled, and the virus is released when the cells are shed. So, HPV uses the normal maturation of epithelial cells to complete its life cycle.
E5 is the earliest protein expressed after infection. It primarily targets the cell's internal membrane system, including the endoplasmic reticulum, Golgi apparatus, and endosomal membranes. This disrupts normal intracellular trafficking and creates an environment that favors viral replication. One of E5's key functions is to enhance epidermal growth factor receptor, or EGFR, signaling by preventing the receptor from being degraded. This causes continuous activation of the MAPK, ERK and PI3K, ACT signaling pathways, leading to increased DNA synthesis, enhanced cell proliferation, delayed keratinocyte differentiation, and continued replication of HPV DNA. For altered endosomal trafficking, in HPV-infected cells, E5 interferes with the pathway, so receptors are not broken down efficiently. As a result, receptors keep cycling inside the cell instead of being properly removed. Because of that, it allows growth signals to continue for a longer time than normal. When the cell cannot shut off these signals, it becomes more likely to survive, proliferate, and undergo abnormal structural changes. For increased EGFR signaling, when E5 present, degradation is inhibited, so EGFR remains active on the cell surface for longer periods. This prolonged signaling stimulates cell proliferation and cell survival. It creates favorable environment for HPV replication. For vacuole formation, E5 causes cytoplasmic vacuoles to appear because it disturbs ion transport, membrane permeability, and vesicle fusion. These changes lead to enlargement of endosomes and accumulation of vesicle-like structures inside the cell. For membrane alteration, E5 is a hydrophobic transmembrane protein, meaning it can insert itself into intracellular membranes. Once inserted, it changes the physical properties of the membrane, including thickness, curvature, and stability. It can also disrupt vesicle transport, which further affects normal cellular trafficking. When these systems are disturbed, the cell develops the abnormal vacuolated appearance and altered growth behavior characteristic of HPV effect.
There are also other proteins that also play roles in the formation of koilocytes, are causing the changes to the infected HPV cells. For E4 proteins, it mainly affects the keratin intermediate filament networks. E4 proteins are mainly expressed as E1-E4 proteins, which is abundant at the late stage of infection. It also causes G2 arrest, which is the stage where cells are producing proteins in growth for cell division. E4 proteins works by reorganizing the cellular keratin network through direct keratin binding, and causing keratin phosphorylation and ubiquitination, where this process marks the keratin for proteasomal degradation, although it is also found to reduce the proteasomal function. This interaction with keratin causing the keratin depletion as it can be seen when moving closer to the cell surface. For E6 protein, it targets the p53 tumor suppressor found in the cell regulation system. However, on its own the protein cannot target it for proteasomal degradation, hence requiring cellular E3 ubiquitin ligase, binding through its LXXLL motif, and form a complex that can label and cause the proteasomal degradation. Other than that, it also causes continuous telomerase activity of the cell, which usually ends when reaching senescence. But the protein prevents the cell from stopping the telomerase activity, causing continued cell proliferation by binding to the HTERT catalytic subunit of the telomerase enzyme. E6 protein also interacts with PDZ domain proteins, which are found on the cell junctions for cell communication through signaling complexes, which also affect the cellular polarity. Overall, E6 proteins work by contributing to cell apoptosis and replicative immortality. E7 proteins target PRB, which is also a tumor suppressor or retinoblastoma tumor suppressor. When binded to PRB, it prevents the PRB from interacting with E2F transcriptional repressor, causing the factor not be able to bind to the E2F sites on cells, causing continuous cell cycle and S-phase progression. Next, it also interferes with CDK inhibitors, which cause the cell cycle progression and prevent DNA damage-induced cell cycle arrest, while also affecting HDAC complex activity, which causing E2F factor increased production, thus disrupting YHE transcriptional complexes and cellular proliferation. In normal cells, the centriole usually undergoes one round of duplication for each cell division. But in E7 protein-affected cells, there are abnormal centrosomal duplications, producing multiple centrioles for each cycle. Overall, E7 protein causes cells maintain a replication state where the unscheduled DNA synthesis causing this core muscle instability. For increased EGFR signal, when E5 present, degradation is inhibited, so EGFR remains active on the cell surface for longer periods. This prolonged signaling stimulates cell proliferation and cell survival. It creates favorable environment for HPV replication. For vacuole formation, E5 causes cytoplasmic vacuoles to appear because it disturbs ion transport, membrane permeability, and vesicle fusion. These changes lead to enlargement of endosomes and accumulation of vesicle-like structures inside the cell. For membrane alteration, E5 is a hydrophobic transmembrane protein, meaning it can insert itself into intracellular membranes. Once inserted, it changes the physical properties of the membrane, including thickness, curvature, and stability. It can also disrupt vesicle transport, which further affects normal cellular trafficking. When these systems are disturbed, the cell develops the abnormal vacuolated appearance and altered growth behavior characteristic of HPV effect.
To understand how a koilocyte forms, we have to look at what happens immediately after human papillomavirus infects the basal epithelial cells. Once inside, the virus hijacks the host machinery to express its early proteins, specifically, the E5 and E6 oncoproteins. These proteins act as the primary drivers of cellular remodeling. Keratin disruption. Normally, a dense network of cytokeratin filaments acts as the internal scaffolding of the cell, maintaining its shape. However, the E5 and E6 proteins destabilize this network. They cause the cytokeratin filaments to collapse and aggregate, pushing them away from the center and bundling them tightly against the cell's outer periphery. Because the cytokeratin scaffolding has collapsed and migrated outward, it leaves behind a massive structural void in the center of the cell. This physical empty space manifests as a large cytoplasmic vacuole. This empty vacuole wraps entirely around the nucleus, creating the classic paranuclear halo that we see under the microscope. The displaced cellular contents form a condensed, dense rim at the very edge of the cell, sharply outlining this clear halo. At the same time, the nucleus undergoes profound changes. Due to the viral E6 protein disrupting cellular stability, the nucleus swells to 2 or 3 times its normal size. The chromatin inside becomes hyperchromatic, meaning it stains much darker and clumps irregularly. As the expanding vacuole presses against this altering nucleus, the nuclear membrane wrinkles, creating a distinct, raisin-like appearance. So combining all of these features, the peripheral cytokeratin collapse, the clear paranuclear halo, and the enlarged, wrinkled nucleus, you get a fully formed koilocyte. In clinical practice, recognizing this specific cellular transformation in Pap smears is our definitive cytopathic hallmark for diagnosing an active, productive HPV infection.
First, let's look at what koilocytes are. Koilocytes are squamous epithelial cells that have been infected by human papillomavirus, or HPV. They represent the cytopathic effect of HPV, which means these are visible structural changes caused by the virus in infected cells. HPV does not immediately kill the cell, but instead alters its structure and growth while the cell remains viable. Koilocytes are commonly identified during Pap smear examinations and in histological tissue sections, especially in the cervical transformation zone, where HPV actively infects maturing squamous epithelial cells. On the image, you can see a normal squamous cell on the left and a koilocyte on the right. Their presence is considered a hallmark of HPV infection, especially in low-grade squamous intraepithelial lesions, or LSIL. Next is the perinuclear halo, which is the most characteristic feature of a koilocyte. It appears as a clear, unstained area surrounding the nucleus. This is not empty space, but a result of cytoplasmic vacuolization and displacement of organelles. This halo forms after HPV infects the squamous epithelial cell. The virus produces F5, E6, and E7 proteins which disrupt the keratin intermediate filament network around the nucleus. As a result, the cytoplasmic organelles become displaced, causing cytoplasmic vacuolization. The remaining cytoplasm is pushed towards the edge of the cell, creating the characteristic clear halo around the nucleus. Because of this, the perinuclear halo is considered an important cytopathic effect of HPV and is commonly seen in cervical cells infected with both low-risk and high-risk HPV types. Besides the halo, koilocytes also show several nuclear changes. First is the hyperchromatic nucleus, where the nucleus stains darker than normal because of increased chromatin density. This results from HPV-induced abnormalities in DNA replication and indicates cellular atypia. Second is nuclear irregularity. Instead of having a smooth outline, the nuclear membrane becomes wrinkled or irregular due to disruption of the normal nuclear structure, reflecting dysplastic cellular changes. Lastly, the nucleus becomes enlarged, usually about 2 to 3 times larger than a normal squamous cell nucleus. This is caused by increased DNA content and abnormal cell cycle progression, resulting in an increased nuclear to cytoplasmic ratio, which is another feature of dysplasia. Together with the perinuclear halo, these nuclear changes strongly support the diagnosis of HPV infection. The final characteristic is binucleation, which means a single squamous epithelial cell contains 2 nuclei, and these nuclei are usually similar in size and shape. This occurs because HPV disrupts the normal cell cycle. Although the nucleus divides normally, the cytoplasm fails to separate completely during cytokinesis, leaving one cell with 2 nuclei. While binucleation alone is not specific to HPV and can occur in other conditions like inflammation, it becomes an important diagnostic feature when it is observed together with the perinuclear halo, hyperchromatic nucleus, nuclear enlargement, and irregular nuclear membrane, which strengthens the diagnosis of koilocytosis.
Now, I would like to explain the clinical significance of koilocytosis and conclude our presentation. First, koilocytosis is considered a marker of productive HPV infection. This means that when koilocytes are seen under the microscope, they indicate that HPV is actively replicating inside the epithelial cells. Therefore, the presence of koilocytes is one of the most important cytopathic effects of HPV infection. Secondly, koilocytes are commonly associated with low-grade squamous intraepithelial lesions, or LSIL. They are one of the key microscopic features that pathologists and cytologists look for when examining cervical cytology samples. Their presence helps support the diagnosis of HPV-related cellular changes. However, it is also important to remember that if the infection is caused by high-risk HPV types, such as HPV-16 or HPV-18, and the virus is not cleared by the immune system, the infection may persist. Persistent HPV infection increases the risk of disease progression from LSIL to high-grade squamous intraepithelial lesions, or HSIL. Without proper follow-up and treatment, HSIL may eventually progress to cervical cancer. For this reason, koilocyte identification is also very important in cervical cancer screening. During Pap smear examination, identifying koilocytes allows healthcare professionals to detect HPV-related abnormalities at an early stage. Early detection gives patients the opportunity to receive appropriate monitoring or treatment before the lesions become more severe. Why is HPV research important? HPV is one of the major causes of cervical cancer and is responsible for most HPV-related cancers worldwide. By understanding how HPV infects cells and how its viral proteins affect normal cellular function, researchers can improve diagnostic methods, develop better treatment strategies, and strengthen prevention programs. In addition, combining HPV vaccination with regular cervical screening has been proven to significantly reduce the incidence of cervical cancer and improve women's health worldwide. To conclude, HPV infects the basal cells of the cervical epithelium and expresses the viral proteins E4, E5, E6, and E7. These proteins disrupt the normal functions of the host cells and eventually lead to the formation of koilocytes. Since koilocytes are the hallmark of productive HPV infection, recognizing them in cervical cytology is very important for the early detection of precancerous lesions. This allows timely clinical management and plays an important role in preventing the development of cervical cancer. That is all from our group. Thank you very much for your attention.